Hi, good afternoon, everybody. And it's a real pleasure and an honor to welcome you to today's seminars in oncology. And we're really thrilled to have as our guest today, Dan DeRoche from the Lunenfeld Tannenbaum Research Institute in the Mount Sinai Hospital in Toronto, Ontario. And Dan has been, I think, as many of you know, one of the world's leaders in the understanding of DNA repair. And it's a real pleasure to have him here today. What you probably don't know, and I just heard, is that he had a well-known American relative, Leo Durocher, the long-term manager of the and coach of the Los Angeles Dodgers, and one of the leading coaches in American baseball. So it's a distinctive connection. Two men who know how to make an impact and know how to win. So the other thing I just want to say on a sadder note, I'm actually filling in as host today, and David Livingston had originally invited Dan to speak today, and David, of course, was a giant in the field of understanding of DNA repair, and as well as a mentor to and friend to so many of us. And I cannot say how much we all miss him here today, and he would welcome and regale Dan in a very special way if he were here, probably using his French Canadian accent, if he could, and talking about his least favorite hockey team, Les Habitants, the Canadians of Montreal. So this being said, it's just, as I say, a real pleasure and honor to welcome Dan DeRoche. I wish that David were here himself to do so, but we're really glad to have you speaking to us. Thank you, Dan, very much in advance.
Thank you, Matt, and thank you for doing this such short notice. You stole my thing because David used to always talk to me in French and always talk to me about the Montreal Canadiens, and he was such a gentleman. He's sorely missed. So I will share my screen, and what I will talk to you today is two stories. Where we basically have used CRISPR-based synthetic lethal screens to basically try to understand genome instability pathways, and at the same time identify some potentially tractable vulnerabilities. And obviously I want to dedicate this presentation to David. He's been a real giant in the field, and he was actually very kind to me over the years, and a very it was amazing to talk to him every time we met. So thank you to him, and thank you also for forming so many people in the field that are now doing amazing. So just to put everybody on the same page, I just want to introduce you a little bit about the concept of synthetic lethality. So synthetic lethality is a type of genetic interactions that was described in the mid to early 20th century. And it's a type of genetic interaction that refers to two alleles of genes when on their own, these alleles, these mutants, are viable, but when they're combined, they produce either cell or organismal lethality. And when we think about this idea, this is actually very relevant in oncology, because if you think of cancer cells as cells that accumulate mutations in their genome, there's great potential for uncovering synthetic lethality in genetic interactions. And as first really conceptualized by Lee Hartwell and Stephen Friend, if we think about the idea of an inhibitor of a gene product, in this case, protein B, as an idealized mutation, we may be able to reconstitute or reimpose synthetic lethal interactions in cancer cells, in this case, because gene A is mutated and displays a synthetic lethal interaction with gene B. And this would lead to lethality specifically of the cancer cells. Obviously, this makes it very attractive as an approach in oncology. And there's also other features that makes it very interesting. That's why we're interested in it. First of all, it actually can target other types of genetic alterations than oncogenic mutations. It also might be active against mutations independently really of the tumor types, although this I think remains to be seen. And what I think is more important is that over the years, cancer genome sequencing has given us a list of potentially actionable mutations for which we can try to develop synthetic lethal-based drugs. But I think most important for me is that synthetic lethal interactions can uncover interesting biology. And we have great examples of this in model organisms, specifically in budding yeast. And this is what really attracts me in this space, trying to understand biology of genome maintenance pathway. So regarding synthetic lethality in oncology, another important point I want to make here is that high-grade serous ovarian carcinoma actually is a very good model to consider synthetic lethal approach. And first of all, is because the best example is used to treat a fraction of tumors in ovarian cancer tumors. And this is because it's a cancer type that has high levels of genome instability. And it's kind of roughly divided by around 50% of tumors which harbor deficiencies in homologous recombination genes such as BRC1, BRC2, and the RAD51 paralogs. But also, and for those, we have a synthetic lethal therapy, but also there's another large portion of these tumors that display amplification of the cyclin E genes, which also impair genome stability. And these actually tumors are highly chemoresistant and a very bad prognosis. There's very few actionable oncogenic drivers in this tumor, so really that makes it very important setting to consider synthetic lethal approaches. And as I mentioned, really this has been, highly stimulated by the development and subsequent approval of PARP inhibitors for the treatment of breast and ovarian cancer, where there are germline or genetic alterations in the BRC1, BRC2 genes. And this really has established the synthetic lethality proof of concept. And really, oops, sorry, I have a very sensitive mouse. The idea here is that in normal cells, the poly-ADP ribose polymerase enzymes, PARP, acts to repair lesions that are usually innocuous single-strand breaks that originates, for example, from the repair of base damage. So these lesions are innocuous because PARP inhibitors PARP can repair them, and in the absence of PARP, there are backup mechanisms such as homologous recombination that can repair this. So in normal setting, treating normal cells with PARP inhibitors is pretty innocuous. However, in cells that have undergone a biallelic loss of BRCA1 and BRCA2, basically we've lost this backup pathway of BRCA1 and BRCA2, and inhibiting PARP leads to the accumulation of lesions that are lethal. Sorry, I'll try not to use my mouse too much. So basically, this has been a great proof of concept, but also raises the question, whether this is a black swan event or we can actually identify other synthetic lethal interactions of therapeutic importance. There's been others that have been discovered, some of them by members of the audience, but really we wanted to go and revisit this question. And basically we want to ask the question, what actually is the best synthetic lethal interactions with BRCA1, BRCA2 loss? Obviously, this is a question that obviously is difficult to answer because it really depends on everybody's opinion. But here I will define it as a genetic interaction that would be highly penetrant, that would be irrespective of cell type or the type of mutations in the BRCA1, BRCA2 genes, that is really strong phenotype, and also that is differentiated for PARP inhibition, because we don't want to just rediscover PARP here. And to do so, we enlisted this team here. Primarily, this was the work of Salome Adam and Sylvia Rossi, two postdoctoral fellows in the lab, with the help of many people in my lab. And as you will see later, it's a collaboration with an old friend of mine, Manuel Stucki, from the University of Zurich, and his student, Mara DeMarco Zumpit. And we also had help from Repair Therapeutics, which is a company I co-founded. So that should serve also as a conflict of interest statement. So the approach we took were isogenic CRISPR genome scale screens, dropout screens. And essentially what we did is basically we generated an isogenic BRCA1 deficient cell line in RPE1 cells and used a second cell line, BRCA2 deficient cells that we can obtain commercially that was done in DLD1 cells. So RP1 and the other one are two different cell backgrounds. One is a retinal pigment epithelium and the other one is a colorectal cancer cell line, which had basically the two main genetic alterations in HR deficiency, BRCA1 and BRCA2. So we do these screens by infecting the two isogenic cell lines with a guide RNA library. In this case, we're using the TKO libraries developed in Toronto, either the V2 or the V3, which is the newer generation. And essentially what we do is we culture these mutated pools of cells over time. And at the endpoint, we try to identify guide RNAs that lead to selective lethality in the BRCA deficient setting. And we did these screens in two different backgrounds and two different genes, so we can identify common hits that may be universal or highly penetrant synthetic lethal genetic interactions. So these are the results of the screen. These were analyzed by a algorithm called CCA, which was developed by Repair Therapeutics. And shown here, highlighted in blue, are the 10 common genes that were identified in both screens. Obviously, there's screens that are private to each of these screens and, as well. But we were focusing on the common hits. And satisfactorily, we identified PARP1, which I think was very good to see, and also identified other genes that over the years were also shown to be synthetic lethal with BRCA1 and BRCA2, including CHT1L, which is a nucleosome remodeler, XRCC1, and also the APEX2 gene, which codes for the APE2 nuclease. So we wanted to know which gene here was the gene we should work on. And to do this, we decided to basically look at a resource that many of you will be familiar, which is the cancer dependency map. The cancer dependency map, as you know, is basically resources where genetic dependencies in tumor cell lines are basically being mapped, which allow us to basically ask the question in that setting, if we identify the set of cell lines that are biallelically mutated in BRCA1 and BRCA2, which are the genes that are selectively essential in that setting. And lo and behold, in that analysis, the genes that displays the strongest vulnerability in BRCA1, BRCA2 deficient cells is this gene at the top right called CYP2A. And PARP1 was also positive in this analysis. So this is interesting, and we decided to go and validate this genetic interaction between CYP2A and BRCA1 and BRCA2. Or as you can see here, guide RNAs. I'll take this. Sorry, I'll take the laser pointer. That's going to help me. Loss of CYP2A leads to very profound lethality in both BRCA1 and BRCA2 settings, and we can complement this defect with a guide RNA resistant transgene, CYP2A transgenes, really proving that this genetic interaction is caused by the loss of CYP2A. So, so this is a strong interaction seen in different settings and also seen in tumor cell lines. So really begging the question of what is CYP2A? Well, a bit is known about CYP2A. It's a gene that originally was found as a protein that was overexpressed in various malignancies, and also that was associated with poor outcomes. Was shown to modulate the function of PP2A and the c-MYC oncogene. It's not an enzyme. It basically contains an N-terminus highly structured core formed of armadillo repeats and also forms a dimer, followed by a long and conserved cortical domain. However, CYP2A had not, especially at the onset of this work, been associated with DNA repair. So we really, the challenge at this point was to really trying to see what CYP2A was doing. So we considered two type of models. First is what we call the endogenous lesion repair. And this is the model really that applies to PARP and APE2 as well. So this is a model where PARP and APE2 are enzymes that prevent the accumulation of endogenous DNA damage that needs to be repaired by HR if these enzymes are not there. So that makes sense. You inhibit PARP, you accumulate lesions that require BRCA1 and BRCA2. But there's also a number of other possibilities, and one of them that we're quite interested in is the idea of adaptive mechanisms. We know that loss of BRCA1 and BRCA2 in cells is actually, in most cases, lethal, leading to a really high level of genome instability. So in the evolution of BRCA1 and BRCA2 deficient cells, these cells must adapt to the loss of BRCA1 and BRCA2. And we know actually p53 loss is one of these adaptive mechanisms, but there's likely others, and these might represent, alternative means of maybe targeting this deficiency. So we decided to test this idea. And first, we wanted to ask the question, is the loss of CYP2A causing DNA lesions and spontaneously, and whether these lesions engage the HR pathway? And we first looked at this using the accumulation of RAD51 subnuclear foci by immunofluorescence. And this is really a proxy of the engagement of the HR pathway, of DNA lesions undergoing HR. So we looked, and these are in spontaneously, not after DNA damage. And what we see is that loss of CYP2A does not increase the load of DNA lesions engaging HR in these cells, suggesting that this is not what CYP2A does. It's not preventing the accumulation of endogenous DNA lesions. And we strengthened this by looking at a type of HR reaction called sister chromatid exchanges. And we know that sister chromatid exchanges are upregulated in response to agents such as PARP inhibitors, olaparib. And what we found is that CYP2A loss is not affecting the basal sister chromatid exchange numbers or the DNA damage induced numbers, really suggesting that CYP2A acts in a way that is very distinct from PARP inhibition and also APE2 function. So one clue came when we were surveying the various assays that we have at our disposal, and one assay was the formation of micronuclei. So again, I think this audience, many of you would be familiar with the work of David Pellman, who's shown that micronuclei are an important source of chromotripsis in cancer genomes. So micronuclei forms when chromosomes or chromosome fragments are missegregated, and where a new nuclear envelope forms against this missegregated genomic mass. And there's really two flavors of micronuclei. One flavor, which is caused by the loss of whole chromosomes, and there's another flavor caused by broken chromosomes. And these can be easily distinguishable because you can stain for kinetochores or centromere themselves. And basically, acentric fragment loss basically is kinetochore negative. So we examine micronucleation in BRCA1 and BRCA2 deficient cell, just showing you BRCA2 deficient cell here. When we depleted CYP2A, and what we saw is a really striking increase in micronucleation. And basically, this micronucleation was exclusively eccentric. So basically saying that loss of CYP2A caused an accumulation of broken chromosome fragments in these cells, okay? And this was very striking. It was only in the BRCA deficient setting. We actually think that the micronucleation is due to a problem in DNA replication. And I won't show you all the data, but this is one piece of evidence that we have, is that if we treat a normal cell, so these are BRCA wild-type cells, either with a DNA replication inhibitor, afidecolin, or ATR inhibitors, which also perturbs DNA replication, we see also a very striking increase in micronucleation in CYP2A negative cells. And actually, I'm going to skip this because actually it shouldn't have been there, but I missed my slides there. But okay, so what I wanted to say is that DNA replication stress caused micronucleation in CYP2A cells, and just pretend you're not seeing the right side of this slide. So this is kind of at this point, the model that we have is what we think is happening is that in the absence of BRCA1 and BRCA2, we're having incomplete replication. And there's a lot of data to support this part of the model. And we know that incompletely replicated DNA needs to be taken care of in mitosis. And what we think is happening is that CYP2A is either acting in the repair of chromatin breaks in mitosis, or actually acting to promote the accurate segregation of broken chromosome fragments in mitosis. And our data suggests that the micronucleation that ensues from the loss of CYP2A is responsible for cell death. But obviously, we need to gain a little more mechanistic insight about the workings of CYP2A. And we asked the question whether it was a DNA repair protein. So if it was a DNA repair protein, one would probably expect that it would work in the nucleus. However, loss of CYP2A in cells, actually CYP2A is a cytoplasmic protein, okay? And this staining is very specific because it's completely gone in the CYP2A knockout cells. This is not, fully consistent with a DNA repair factor. So we thought maybe that irradiating the cells would cause a translocation of CYP2A from the cytoplasm to the nucleus. So we asked this question. So we irradiated cells, we can induce DNA damage in those cells, but no dice were not able to induce the translocation of cytoplasmic CYP2A into the nucleus in those cells. However, in our fields, we did see some cells with foci of CYP2A, so subnuclear dots that co-localize with a DNA damage marker, gamma H2AX, okay? And these cells, were around 1 to 3% of the population, and really it doesn't take a PhD in cell biology to know that it's actually a mitotic cell, okay? So it turns out that CYP2A can localize to sites of DNA damage, specifically in mitotic cells. And so this is an example here. If we radiate cells and specifically look at mitotic cells, we see a dose-dependent increase of CYP2A foci in these cells. And furthermore, if we looked at BRCA deficient cells in mitosis without any exogenous DNA damage, we see an increase of CYP2A. We see actually an accumulation of CYP2A foci in those cells, really suggesting that this mitotic DNA damage localization of CYP2A is relevant to the CYP2A BRCA synthetic lethality. So in the interest of time, I will skip some work, but what I want to tell you is that we find that CYP2A forms a protein complex with another genome maintenance factor called TOP-BP1. So TOP-BP1 is a DNA repair or DNA damage adapter involved in multiple processes in the cell, including DNA replication and ATR activation. And actually, CYP2A and TOP-BP1 localize with each other on mitotic DNA lesions. And what I will skip also, but summarize here is that this CYP2A top BP1 complex basically is downstream of what appears to be two independent pathways. One pathway is dependent on this other adapter protein called MDC1, and this responds to DNA double-strand breaks, such as those caused by ionizing radiation. But there's another pathway that leads to top BP1 and CYP2A localization that seems to be responding or that is responding to under-replicated DNA, and this pathway is independent of MDC1. And this just to show you a little bit of data to show you this really intimate partnership between CYP2A and top BP1. So these are cells that are being co-stained with CYP2A and top BP1 antibodies. And as you can see, there's a perfect overlap between the CYP2A and top BP1 signals. So this is data from Mara in Manuel's lab, but we have the same data in the lab, but maybe not looking as nice. So CYP2A and top BP1 localization depend on each other and perfectly overlap only in mitotic cells, not in interphase cells. So MDC1, which is important for that double-strand break repair, this double-strand break dependent localization pathway is, strangely enough, not important for the viability of BRCA-deficient cells. So these are experiments where we've knocked out MDC1 and CYP2A, and we're not seeing an effect of depleting MDC1 in BRCA-deficient cells, although we see the effect of CYP2A, really suggesting that it's this other pathway that is important for maintaining viability of BRCA-deficient cells. And this is consistent with the fact that BRCA-deficient cells accumulate under-replicated DNA in mitosis. And this is data, for example, from Jason a few years ago. So that begs the question whether that interaction between CYP2A and top BP1 was important to maintain the viability of BRCA-deficient cells. And to do so, first, we needed to map the site of the interaction. And using a variety of approaches, we converged on a CYP2A binding region on top BP1, which is located between the BRCT5 and 6 domains. So top BP1 is a multi-BRCT domain containing proteins. We even identified residues that are necessary for this interaction. So we can generate mutants now of top BP1 that disable the interaction with CYP2A. And this was important because top BP1 is an essential gene, so we can't really just knock it, when we knock it out in both wild type or BRCA deficient cells, I'll show you an image in a minute, you see that it leads to lethality in both settings. So that's not very informative to see if it's working with CYP2A. So we needed to do a separation of function mutant. And we can do this separation function mutant either by deleting this region or making this AAA mutations in the binding region. And, so when we generate these mutants, then we first ask whether we lost the localization to DNA damaged sites in mitosis. And as this is basically our gene replacement experiment where we deplete wild type top BP1, and these cells are reconstituted with a guide RNA resistant top BP1 transgenes that is either wild type or that cannot bind CYP2A. And as you can see here, we lose the localization of both top BP1 and CYP2A when these two proteins cannot interact, really suggesting that this complex needs to be together to localize to mitotic DNA damage. So, but this also allowed us to really ask the question, is this interaction important for the viability of BRCA deficient cells? And you may want to focus on the right side because the left side is the quantitation, but it's more visually clear on the right side, is what the experiment says. Depleting top BP1 in both wild type and BRCA deficient cells leads to lethality, as I just told you. This is because top BP1 has other functions that are essential, such as regulation of replication initiation. If we complement these cells with wild type top BP1, we can restore the viability of these cells. But if we introduce a top BP1 variant that can no longer bind with to CYP2A, what you see is that you get selective lethality in the CYP2A in the BRCA deficient cells, really suggesting that this interaction is essential for viability of BRCA1 deficiency, BRCA deficient cells. We've done it with BRCA2 and BRCA1. And I just want to say that this is also accompanied by micronucleation, really suggesting it's this lethal genome instability that is causing the cell death. Although exactly how this is done is not clear. So obviously, in thinking a little bit about translation, we wanted to really ask the question next, if we could find a way to disrupt this interaction in cells, what would we see? And this was greatly helped by serendipity, because Salome found that if she expresses a short fragment of TOPBP1, which contains the CYP2A binding region, she can actually basically have a very, very potent competitive inhibitor of the interaction. And when she places this fragment, which we call B6L, it means BRCT6 left, when she places this fragment under the control of an inducible system where she can induce the expression of B6 cells using a small molecule called SHIELD1. So this is a system that Tom Wandless at Stanford has developed. She can basically induce expression of B6L. And what you can see here is that we rapidly lose the ability of CYP2A and TOPBP1 to form mitotic DNA damage foci. Okay, so this is giving us a great tool to kind of acutely disrupt the interaction between TOPBP1 and CYP2A. So this allowed us to test whether we can, what happens if we disrupt the interaction in either wild type or BRCA deficient cells. And what you can see here is that we get a very selective inhibition of proliferation only when we induce the expression of this B6L competitor peptide. And this is quantitated in 3 experiments here. And what I'm not going to show you is that this growth arrest is completely reversible. So if we take these cells and remove the SHIELD1 compound, these cells are no longer able to grow. They're basically, we get no clonogenic potentials from these cells, really suggesting that these cells are terminally, have stopped proliferation or even have died. So, so this really made us think that we should test whether or not we can control tumor growth with this system in vitro to see what happens in the context of a tumor. So we teamed up with the team at Repair Therapeutics to do this experiment. So the first thing they did was to basically look at the pharmacokinetic properties of the SHIELD1 compound. And in fact, there's a more water-soluble version of SHIELD1 called Aqua SHIELD1 or AS1 here. And what they found is that it's actually not a very bioavailable compound, and the best they could find is that they could reach a concentration in the plasma above IC50 for the interaction for a maximum of 4 hours a day. Okay, so if any of you have done some early phase drug development, you'll know that this is not great, inhibiting your target only for 4 hours a day. But they decided to go ahead with the experiment in mice, and they looked at the impact of disrupting this interaction basically periodically for only a few hours a day. And what you can appreciate on the right is that we're able to have a great tumor growth inhibition in this setting, up to 85%. And this is done only with periodic inhibition. So periodic inhibition of CYP2A TOPBP1 interaction was sufficient in this tumor setting to induce profound tumor growth. And we're really hoping that if we're able to cover the target or inhibit this interaction for more than a few hours a day, we would be able to have regression. And this is something we'd like to work on in the future. So what I showed you in this part of the study is that CYP2A and TOPBP1 seems to play a role in preventing the centric chromosome misaggregation. And we'd like to think that CYP2A and TOPBP1 may in fact act as a bridge that keeps eccentric chromosome fragments attached to the kinetochore bearing chromosome, the centrosome containing chromosome during mitotic cell division. And there are other reasons why we favor this model. But what I've also told you is that this is a really highly penetrant synthetic lethal interaction, and we're trying very hard to find ways to possibly identify inhibitors of this interaction. Obviously, that also raises a number of other questions. What is actually upstream and downstream of this complex? And these are areas of investigation in the lab. But clearly, what I hope I've convinced you is that this mitotic DNA damage response pathway in mitosis by TOBP1 and CYP2A is really important in BRCA deficient cells. And I just want to mention that actually the paper has just been published today in Nature Cancer. So, and I'm sure it's probably better explain the results than I just did in the past 20 minutes. And also there's another paper from Manuel's lab, currently still in preprint form, that talks about the MDC1 angle of this pathway. So in the next 15 minutes, I want to talk to you about another synthetic lethal story, which is really our attempts to identify genetic vulnerabilities against the amplification of the cyclin E oncogene. And this is work that was initiated by Jordan Young when he was a PhD student in my lab, and actually he's moved since then to Repair Therapeutics, where he's heading the functional genomic platform there. And this work also has been hand in hand with a postdoc in my lab, Dave Gallo, who's now also with Repair. And this is kind of a theme in my lab. Repair tends to steal all my people. And but actually he's done a great work there. And as you'll see, it was a very close collaboration also with Repair Therapeutics and many members of that team. So cyclin E, as I mentioned earlier, is cyclin E amplification is found in approximately 20% of high-grade serous ovarian carcinoma, but it's also found in a number of other tumor types where it is also associated with really poor prognosis and really aggressive diseases. At the time we started this work, there was no available selective CDK2 inhibitors. So we thought that a synthetic lethal approach might be actually feasible to do in the cyclin E amplified setting. So what we know from cyclin E amplification is that obviously cyclin E amplification leads to an increase in the expression of the cyclin E protein, and that leads to the activation of cyclin E CDK2 complexes. And the cyclin E CDK2 complex is very important for the orderly progression of cells through the G1S transition. And under conditions where you have cyclin E amplification, some of the hallmarks that are observed are unscheduled entry into S phase. This is associated with DNA replication stress, genome instability, but also genome doubling. And we actually thought that these phenotypes might be actually targetable using a synthetic lethal approach. So we decided to develop a model, our own model of cyclin E amplification. And actually, the idea here was to do a model where we could have chronic high levels of the cyclin E proteins. There's actually a lot of models in the literature where you have an acute and transient overexpression of cyclin E, and we didn't think that was a good reflection of what was going on in tumors. So we generated cell lines using a piggyback system, again, in our workhorse RP1 cell line. And we generated a number of cyclin E overexpressing clones. We'll call these cells cyclin E or CCNE high. CCNE is the gene for cyclin E. And this was associated with a lot of the common hallmarks of cyclin E amplifications, such as DNA replication stress, premature entry into S-phase, problems with replication licensing, etc. So we screened these two clones. In fact, they have different growth properties. So we felt it would be safer to go with independent clones. And basically, these are the results of the screen here. Again, because they're done in these two cell lines, we actually focused on what was common in both cell lines. And these are, again, the same kind of strategy we used. These are the genes that were common, that are highlighted here. And this is how they came in different screens. So we basically had a number of genes associated with anaphase promoting complex. We had the RNase H, 2 of the 3 RNase H genes. A few other genes that are associated, such as this one, as DNA replication stress. We have FBXW7, interestingly, which is actually a negative regulator of cyclin E. And then we had this gene here, which was the top hit, which was PKMYT1, which will be the focus of the next few slides. Just like we did for CYP2A and BRCA deficiency, we also mined the depth map, basically to identify which of these genes that were identified in the isogenic setting were also hits in the depth map data. This is what we see here, is that in the depth map data, obviously we see very strong signal for cyclin E and CDK2, but also we see a very strong signal for PKMYT1. So PKMYT1, as showed you, also was a top hit in the isogenic setting, really suggesting that PKMYT1 might be an interesting gene interaction to follow in cyclin E amplification, both in the tumor settings and obviously in the isogenic setting as well. So what is PKMYT1? So PKMYT1 is also known as MYT1. It's one of the 2 kinases that inhibits the CDK1 kinase, the other one being WEE1. PKMYT1 is selective for CDK1, and that's going to be important, whereas WEE1 can inhibit both CDK1 and CDK2. And the role of PKMYT1 is to phosphorylate CDK1 on threonine 14 to inhibit it. And that threonine 14 phosphorylation is one of the targets of the CDC25 phosphatases, which are the activators of CDK. PKMYT1 is located in the cytoplasm. Actually, it has a interaction domain that targets it to the cytoplasmic phase of the endoplasmic reticulum. So it's a cytoplasmic inhibitor of CDK1, whereas WEE1 is a nuclear inhibitor of CDK1 and CDK2. So we decided to go and validate this genetic interaction and also ask the question whether or not the catalytic activity of PKMYT1 was involved, because obviously this is important if we're thinking about therapeutics. What we found is that in the isogenic setting, the answer was clearly yes. We could validate this genetic interaction, so we could rescue the synthetic lethality with the wild type PKMYT1, and this rescue was completely abrogated when we mutated residues that impair its catalytic activity. This really then convinced Repair Therapeutics to go ahead and initiate a drug discovery program to identify selective inhibitors of PKMYT1. And they identified a number of compounds over the year, culminating in this compound here called RP6306, which is a highly potent and selective PKMYT1 inhibitor. So it inhibits PKMYT1 in the low nanomolar range. It has very good selectivity against WEE1, and in fact, has only very few other kinases that it touches. It's also selectively inhibits the threonine 14 phosphorylation, as we'd expect in cells. And also it has very good PK properties that indicates that it will be bioavailable as an inhibitor. So with this inhibitor in hand, we wanted to know if we could recapitulate the synthetic lethality in cell lines. So again, looking at our 2 cyclin E high cell lines, and compare to our wild type, you can see that inhibiting PKMYT1 in this setting leads to very profound sensitivity. I must say, so this is, we observed this as well in the RP1 cells, but this is actually is a cyclin E amplification model that we obtained from Dana-Farber, was developed by Ronnie Drapkin, which is basically a fallopian tube cell line. And this is actually highly relevant because the fallopian tube is the cell of origin of high-grade ovarian cancer. And these cell lines were reconstituted to overexpress cyclin E. So the bottom line here is that we're seeing that inhibition of PKMYT1 is very toxic and selectively toxic in the cyclin E overexpressing cells. So then we looked in cancer cells, and what we found is that we actually see a good separation in terms of IC50 from cyclin E amplified cells and cyclin E normal cell lines. We see around between a 10 to 50-fold differences in IC50. So it's not in the range of the PARP BRCA, which is almost 1,000-fold, but it's certainly something noticeable and highly reproducible in multiple cell lines. So what is the mechanism here? So we basically hypothesized that somehow in cyclin E amplified cells, there's an increased dependency on PKMYT1 to hold CDK1 in check prior to the G2 phase. Okay, and this might be there to avoid catastrophic DNA damage, because we know from classic work that entry into mitosis in S phase is actually catastrophic. So we went about and tested this model in a number of ways, and the first one was first CDK1 activated when we inhibit PKMYT1. And to do this, we basically looked at an autophosphorylation site on cyclin B, which is phosphorylated by CDK1 upon entry into mitosis. What we found is something curious, is that indeed we see that inhibition of PKMYT1 by RP6306 leads to activation of CDK1, but this activation is seen only in cyclin E overexpressing cells, which is something that I think is very striking and very curious. At the same time, we also looked at evidence of DNA damage in these cells. And what we saw is similar to activation of CDK1. We see a very strong increase in cells that have a pan-gamma H2AX phenotype. So not the foci, really a pan-nuclear gamma H2AX signal. And this is shown here by cytometry or quantitative microscopy. So, and again, this is completely selective for the cyclin E overexpressing cells. And we see the same phenotype in a tumor cell line, these HCC cells that have cyclin E amplification. So we see this premature entry, this premature CDK1 activity, which is associated with pan-gamma H2AX catastrophic DNA damage. And this phenotype is completely dependent on cyclin B CDK1 activity. So if we deplete cyclin B, we completely suppress the emergence or the induction of DNA damage in these cells, really suggesting that it's really the action of PKMYT1 in restraining CDK1 activity that is at play here. So, so this was our marking model, is that when we inhibit cells, specifically the cyclin E overexpressing cells, we inhibit PKMYT1, we have this activation of CDK1 activity leading to basically a premature entry into mitosis, an unscheduled mitotic entry. And basically we see evidence of this if we look at markers of mitosis. So we look at markers of mitosis in replicating cells here in EDU positive cells. What we see is there's an increase selectively in the cyclin E overexpressing cells in both setting, and we're using two different markers here. Laminase C phosphorylation is one of the early events in mitotic nuclear envelope breakdown. I think alongside this really this mitotic entry, we see evidence of chromosome pulverization, which is really a phenotype characterized by mitotic entry of replicating cells. We see a very large induction of pulverized chromosome when you look at mitotic cells when we treat cyclin E amplified or overexpressing cells with the PKMYT1 inhibitor. So this is really suggesting this idea that cyclin E overexpressing cells have this heightened dependence on PKMYT1 to restrain CDK1 activity and to restrain mitotic entry. So obviously, this agent is now being developed as an anti-cancer agent. So we wanted, Repair wanted to look at efficacy studies in various settings. So this is OFCAR3, which is a cyclin E amplified ovarian cancer cell line. We see activity as a single agent where we get over 80% tumor growth control. But what has been very striking is that when we looked at adding on this agent, adding low levels of DNA replication stress, such as in the case here where we're using gemcitabine, in the same setting, we see a very, very, very profound tumor regression phenotype, really suggesting that this dependency on PKMYT1 may relate to restraining cells from entering mitosis that have a DNA replication stress. So I think that's my last slide here. So we have time for a bit of discussion. What I showed you is that actually we can use isogenic CRISPR-based synthetic lethal screens to discover new targets. I think these are a nice complement to the beautiful DepMap data set that has been generated by the Broad and the Sanger Center. Also in this part of the talk, I showed you that PKMYT1 is synthetic lethal with cyclin E amplification, and that we, meaning Repair, developed RP6306 as a selective and orally bioavailable first-in-class PKMYT1 inhibitor. And cyclin E amplified cells are vulnerable to this unscheduled CDK1 activation. And I didn't really have time to go through this, but we think it's actually due to dysregulation of DNA replication, but also of the FOXM1 MUV-B program, which controls cell cycle gene expression. So RP6306 has now entered phase 1 clinical trial, and I'm pretty sure that Dana-Farber is one of the sites. So we're really looking forward to see what happens here. We hope to help some patients. So I think with that, I thank the people along the way. So I think I can stop here and take a few questions. So thank you very much.
Awesome. So Dan, I will clap for everybody. They would all be clapping for you. And going to start taking some questions from the Q&A section. So the first one comes from somebody with the initials DP and says, "Hi Dan, great talk. I am wondering if CP12 is kept in the cytoplasm because it would somehow interfere with normal DNA replication. Have you tried it to target it to the interphase nucleus?
Yes, actually,
nuclear localization signal."
Yeah, so this is actually a really good question. We actually, Manuel, our collaborator, has tried this experiment. So CYP2A is kept in the cytoplasm, it's exported from the nucleus, so there's a nuclear export signal. And when Manuel mutates it, he can actually drive some CYP2A back in the nucleus. And what he's finding is that, at the gross level, he's not seeing that it's perturbing interphase DNA replication. And this is actually quite surprising. Actually, our expectation was also that it would impede with top BP1 function, which are important in many aspects of DNA replication and ATR activity. So actually, at the moment, our evidence suggests it does not. It's not kept in the cytoplasm for that reason. But obviously, this is early days and preliminary results. So yeah, so we don't know why it's kept in the cytoplasm in mitosis in interface.
All right, awesome, thank you. Thank you, Dan. And that question was from David Pellman.
Yeah,
So
that's
the
what,
next,
what I'm guessing.
so the next question is from Annalisa DeFeo, who says, have you assessed whether the AS1 compound affects PP2A phosphatase activity?
Yeah, so this is actually, is an important question relating to the reported role of CYP2A as a modulator of phosphatase, of PP2A family phosphatase. So we actually have not directly assessed this, but we've done a few experiments to kind of get at this question. And the two experiments were as follows. So the reported role of CYP2A as a modulator of PP2A activity is to be an inhibitor. So if PP2A phosphatases are important, and CYP2A interacts with two adapters of PP2A phosphatase, the B56 adapters, and so if CYP2A acts by modulating PP2A, the prediction would be that inhibiting PP2A would suppress the phenotypes. So to kind of do this, we asked whether the phenotypes of B6 cell expression, so when we added the AS1 compound, were reversed when we depleted B56 proteins, okay, which are the target of CYP2A. And the answer was we didn't see any difference, okay, really arguing that regulation of PP2A activity may not be what's at play here. And then the other experiment we did is we basically look at the interaction of CYP2A and PP2A in cells in our setting. And we have a little trick to do this. And we didn't see any interaction with the CYP2A top BP1 complex. I'm not saying CYP2A does not interact with PP2A, that's been shown, but as part of that complex, it doesn't seem to be interacting. So our evidence suggests that PP2A may not play a role in this biology, at least, you know, not majorly. But it's still an open question. I think an important one. Thank you for the question.
Yeah, great. Thank you. So I'm curious, you identified a number of synthetic lethals in your BRCA1, BRCA2 screen, and there were about 10 genes that scored pretty strongly. One was CYP2A, one was PARP1 itself, and then there were, you know, a number of others that you also identified. APEX2 was one, quite a few others. Can you start to comment on, you know, some of these other genes, and what do you, you know, are you planning to do further studies of those, or perhaps you have? And I'd love to hear your thoughts about those.
Yeah, absolutely. So we have we basically had a preliminary look at most of them. So there's a couple of interesting ones there. In addition to, so APEX2, we've also was first identified by Steve Elledge, synthetic lethal, and we also did some work on it. And what we found is that APEX2 was very important to promote the repair of endogenous ribonucleotides, which are very abundant in S phase. And so it's basically, we can put this one in the bucket of preventing endogenous DNA damage to engage the HR pathway. CHD1L is also a really interesting one. It's actually also been reported by a number of groups recently: Simon Bolton, Roger Greenberg at Penn, and Andreas Lerner in Munich. And what CHD1L does, it's a chromatin remodeling enzyme, or nucleosome sliding enzyme, that plays a role in base excision repair. So it's kind of a helping, or in the same pathway as PARP. In fact, CHD1L deficient cells are extremely PARP inhibitor sensitive. So this one's actually quite interesting because there's a really strong interaction with PARP itself. So we can, you know, so not only you get the effect, the synthetic lethal effect, you also get the sensitization to PARP. And this one, you know, is also an interesting target in oncology. Another interesting one that actually I like quite a bit is the solute carrier. We had SLC 25A28, which basically is a mitoferrin, mitoferrin 2. So it's important to bring iron in the mitochondria, and I think it's part. And my speculation is that it's playing a role in the assembly of iron sulfur clusters, and iron sulfur clusters are really important for DNA repair and DNA replication proteins. So this is kind of a more of a wild speculation, but we see a nice effect there. So these are maybe my most interesting in that list, and obviously XRCC1 acts with PARP, so that also makes sense.
Great, thank you, Dan. Well, Dan, just want to say again, we're just past 5 o'clock, so
Perfect.
It's time to wrap. But just a really great presentation, really pretty fascinating and impactful work. And you know, I just want to say, if David Livingston had seen this talk, he would have come up to you, he'd have put his arm around you, and he would have said just how incredible it was.
All right. Thank you so much, Matt. And thank you for everyone for listening and your questions. Hope I'll see you soon in person. Okay.
We sure hope so. And thank you again for
Bye-bye.
coming to visit in our modern way. All right.
Thank you. Bye-bye.
Take care. Bye-bye.